Well, hello and welcome. Today I'm going to be talking about immunology as it pertains to COVID infection and also something about herd immunity and antigen antibody tests for COVID 19. My name is Dr. Chander Mehta, and I'm a professor emeritus of clinical pharmacology and toxicology. And as far as the immune system is concerned, I will be really very briefly touching on the aspects of immunology that really are more of an overview. But if you want detailed discussion of this very highly complex topic, I'll be more than happy to talk about. So, immune system briefly protects us from outside invaders like bacteria, viruses, fungi, parasites, and also toxic chemicals from some of these invaders. Immune system is as I said, complex system made of different organs, cells, and proteins that all work together. Now, there are two types of immunity. One is present at birth, and it's called innate immunity, and the other one is called adaptive immunity that is acquired after birth. So it is also known as acquired immunity. Now, the innate immunity provides us with the immediate short-term protection against viruses, fungi, bacteria, and also parasites. So this is a short-term protection, very non-specific, just something immediately available. And the adaptive immunity is something that takes time. It's present after birth. Usually after about 6 months, you get very good responses from that. And acquired immunity or adaptive immunity is a long-lasting immunity involving the lymphocytes and other cells of immunity, which I'll mention briefly, but the complex discussion of it, I will leave it up to some other times. Now, antibodies that form part of immunity are Y-shaped molecules, and they combine with the invaders, which we refer to as antigens. So antigen antibody interaction is very important, and COVID virus as an antigen is blocked by antibody. Here is a Y-shaped antibody, and it has a binding site. And this binding site then makes virus unable to penetrate the host cell, and that's very important. If there was no antibody, there was no protection, virus will come, as it shows in this diagram, right in and affect the infection. The binding of the virus to the host cell happens by way of a very important protein called ACE2 receptor protein. This protein is involved in the control of blood pressure and several other functions. So it's a very important site. But somehow virus binds to that. Virus have this hook or spike protein, like pointed here. They bind to the ACE2 site and enter. And when they enter the host cell, they can take over the host cell and produce severe damage. So how do you protect it? Well, you bind the virus with the antibodies. When that is done, they cannot bind to ACE2 protein, and thereby you're protected. Also, some of the immune cells have antibodies that will directly bind, to virus spike protein, and thereby make it unavailable to attack the host cell. So that is another mechanism of antigen antibody reaction. So the best way to produce antibodies is by vaccination. Sooner you get vaccinated, better you have the protection, and prevent hospitalization. Remember, one aim of vaccination is not that you ever get a disease. It is that you should not be hospitalized and put on ventilators and die. And so for that, vaccination has a tremendously good record for almost a year now. Now, you can also use this knowledge about antibodies from immune system to do COVID test. And this COVID test uses this principle of immunology. And here we have two immunoglobulins or antibody that we want to talk about. One is IgG, which is immunoglobulin G, and other one is IgM. As you can see, they are both Y-shaped. And so let's first talk about IgG. IgG appears about 2 weeks after infection, and it lasts a long time, almost 6 months or more. Whereas IgM appears very quickly within a week, so produced 7 days after infection compared to 14 days for IgG. And it stays in blood for about 3 weeks or so, and then what it does really is to fulfill its purpose of providing immediate protection to the infection. So if there is a COVID infection, you find that IgM will be released within a week, and then after about a week, you are really having enough antibody to protect you. Of course, by this time, about 2 weeks later, you have IgG also to protect you. Now, this property of IgM and IgG, this profile, can help us devise a very good immune test for COVID. And what is that? Well, what it is that you have a strip that has 3 main regions. One is a C for control region. Then there is IgG embedded antibodies, and there is IgM antibodies. Now, when you put a drop of blood on the strip, if you have IgG antibodies and IgM antibodies, it will light up this, it will make those positive markings on the strip. So if you have blood that has no antibodies, you are not infected, there's nothing to worry, you are not contagious, and you're okay. You are healthy, but you don't have any antibodies. Now, if you have a situation where you have IgG positive, that means the long-term antibodies are there, but you don't have indication of any short-term infection with IgM being negative. Here you can see that only IgG is positive there. Then you are also lucky here in that you had an infection before, but now you are not contagious or infectious, and you will not be a problem for other people. So that is safe. And so the first 2 cases, no worries, you are okay. But in the next 2 cases, you have IgM as a positive. Let's say the third case, you are IgM positive, and that IgM strip lights up, and you can see that that would indicate that you have in a very short term some infection with exposure to the COVID virus, and you have to be isolated. And when that happens, usually isolation is about week to 10 days, and by that time you are not infectious because you are protected and you are not contagious to other people. And fourth scenario is that you are positive to both immunoglobulins, IgG and IgM. Now, why is that? It's quite possible that you had first a COVID infection and then you got a second time an infection, and that produced IgM, the short-term antibody. So in this case, you are contagious, even though you were exposed before and you were protected then, but not anymore now. So you have to be isolated, and because you are contagious. So this is a very unique way of really um, utilizing our knowledge of antibodies in immunology to devise a test for COVID. Now, many people have asked me about herd immunity, and I like to answer and explain that properly. Herd immunity provides protection, and this is protection for individual person and also for the entire community. So that's why it is herd immunity. If you are a part of the herd, you are protected. So herd could be a neighborhood or a village or a small area where you have people that you mingle with all the time. The herd immunity threshold is very important. You want to find out when you are protected, and that is determined by a percentage which is referred to as HIT percentage. And what you find is if you reach that percentage of vaccinated people or infected people, you are then immune from attacks and you are safe. And this HIT value varies from disease to disease. And for a very contagious disease like measles, the percentage is 90%. So 90% of the community has to be showing antibody protection before you reach the herd immunity. And if you don't reach that level, people who are not vaccinated or infected will get infected. In case of COVID, there was not much data available at first. And so we started out with pure guesstimate and thought it was about 60%. But as more data accumulated, we realized that 70% was a good number to provide herd immunity to COVID. Of course, with the new variants that have come after Delta, the Omicron virus, which is very contagious. In fact, it is even compared to measles. There is a way you can find out how many people are infected by one patient of a disease, and that is referred to as R0 factor. And in case of COVID, starting out, it was about one patient infected 2 or 3 people. And now with Omicron, the result ranges anywhere from 8 to 15 people can be infected. So you can see the infectiousness of the disease has increased. If you want a more detailed discussion about R0 and how it is derived, I can get into that discussion in my future topics or presentations. But suffice to say at this time that for herd immunity to happen, you have to be reaching a critical threshold of percentage of people that are either immune or infected before to provide protection. The best way to explain this is by way of really a diagram. And so I'm going to show this diagram where there are 3 types of population. The blue color patient is not immunized but is healthy. The yellow one is the one that is been immunized either by having a disease or vaccinated and is now healthy. And it's not, and this person is not going to, it's a situation where this person is not going to be contagious. And the red one is a person that has not been immunized and is sick with COVID and is contagious. So let's look at the first scenario. In this neighborhood, this population, everybody is not immunized but is healthy. But these 2 infected people show up in this neighborhood and they infect everybody, almost everybody, and you have high rate of hospitalization and death because there was no herd immunity here, no protection. Second scenario is that you have not reached the threshold, but there are immunized people in the population. However, since it has not reached the critical percentage, let's say for our example, 70% in COVID, you don't have a herd immunity and people will get infected and a lot more people will be hospitalized. Once you reach HIT threshold and you have enough people vaccinated, then you look at the situation where uh, these two patients, when they come into the community, the whole community is protected, except that out of 4 unprotected people, only 3 have no problem. Only one of the, I should say, not only 3, but only one has a problem, and that person is infected. But other 3 out of 4 are fine. So herd immunity here has protected these individuals. So how can we get herd immunity? Vaccination is by far the safest and the quickest way. Getting the disease has a problem in that if you have pre-existing conditions and so forth, you can have severe disease and may even die. So if someone says that I really don't want to be vaccinated because everyone else is vaccinated, it's quite possible that you have not reached the threshold and the community is at danger. And by the same token, if you feel that there is no need to get vaccinated, all you need to do is get a exposure to this, get a disease, and you're okay. Well, you have to be worried about hospitalization and so forth. And lastly, once you reach the herd immunity status, it is not a permanent status. You can still be infected. Example being a condition like smallpox. Now, smallpox is pretty well eradicated from the world, except few pockets like in Afghanistan and Pakistan. Now, if an unimmunized, unvaccinated person goes into that neighborhood, you can easily get smallpox and get severely sick or even die from that. So herd immunity is not permanent protection. You have to be constantly maintain that threshold. And with this, I will end my lecture, and I will like to request you to post your comments on this page and give me your feedback. And any part of this topic that you have questions for, I'll be more than happy to answer that. And also, I have kept an overview and made it very simple for everybody to understand. So if you need more information, I'll be more than happy to have future presentations where I can discuss this thing in much more detail. So thank you again for your time, and I'll see you next time.